Hello students, myself Dr. Shalini Malhotra, and today I will be talking about recombinant DNA technology. Now, first of all, we should know what is gene cloning. Now, DNA cloning is a molecular biology technique that makes many identical copies of a piece of DNA such as a gene. In a typical cloning experiment, a target gene is inserted into a circular piece of a DNA called plasmid. Now, recombinant DNA means when we combine the DNA from two different sources. That two different sources may be one from a plant, other from an animal, one from a plant, other from a bacteria. So we can combine DNA from these two different sources, and then we can insert that DNA into a circular piece of DNA called plasmid. Plasmid is a vector DNA, and this plasmid, once we introduce foreign DNA into our plasmid, it is introduced into the bacteria via a process called transformation, and the bacteria carrying the plasmid are selected using antibiotics. Bacteria with the correct plasmid are used to make more plasmid DNA, or in some cases induced to express the gene and make protein. Now, plasmids, they are circular double-stranded DNA molecules that are separate from the cell's chromosomal DNA. These are the extra chromosomal DNA which occur naturally in bacteria, yeast, and some higher eukaryotic cells exist in a parasitic or symbiotic relationship with their host cell. Plasmids range in size from a few thousand base pairs to more than 100 kilobases. Most naturally occurring plasmids contain genes that provide some benefit to the host cell, fulfilling the plasmid's portion of the symbiotic relationship. For example, some bacterial plasmids encode enzymes that inactivate antibiotics, such as drug-resistant plasmids have become a major problem in the treatment of number of common bacterial pathogens. Now this is the plasmid PBR322. P means small p stands for plasmid. B and R are the names of the scientists who discovered this plasmid, Bolivar and Rodriguez. The size of the plasmid is 4361 base pair. It has an origin of replication. You can see ORI is written, and it has two resistant genes. One is ampicillin resistance, other is tetracycline resistance gene. It has also the sites for restriction enzymes, and by using these restriction enzymes like ecoR1 is a restriction enzyme type 2. As we know that there are three types of restriction enzymes: type 1, type 2, and type 3. Type 1 and type 3, they may cut at a site which is away from the recognition site. Means where we want to cut the DNA, it will not cut exactly at the site, it will cleave away from the recognition site. But type 2 restriction enzymes like ecoR1, from isolated from E. coli, it is the restriction enzyme which recognizes a 6 base pair palindromic sequence. Palindromic means you read from any side, it will read the same, like rotator. You read from both the sides, it will read the same. So ecoR1, it recognizes 6 base pair palindromic sequence GAATTC, and it will cleave exactly where there is a this between G and A site. So this is the structure of a plasmid. It is extra chromosomal DNA, and we can isolate the plasmid. We can we can cut the plasmid and get the foreign DNA inserted into the plasmid. Then we can ligate the two DNAs, and then again we can transform the plasmid. We can transform the host cell with this plasmid. They Now we have another series, PUC vectors. This is the plasmid from University of California. It is quite different from the PBRC22 in the sense that it has a polylinker site. Polylinker means we can use different types of restriction enzymes. Uh, there is a polylinker site that we can use large number of restriction enzymes, and and we can identify, we can say, for example, if you have inserted foreign DNA into this PUC vectors, then and you want to select whether the foreign gene has been inserted into the vector or not, just by seeing the color, blue white selection is there in case of PUC vectors, where in case of PBRC22 we use antibiotic resistance. This is the PUC 19. You can see it has multiple cloning site, means it has the site for, you can use these number of restriction enzymes. Means if you have BAMH1, you can use BAMH1. If you have EcoR1, you have a site EcoR1 also, and it has a LAG-Z gene. LAG-Z gene codes for beta galactosidase, and you have ampicillin resistance gene also, but blue white selection is there. This is the same. Then it has, I have told you this, that we can select the PUC 19, recombinant PUC 19 by seeing the blue white colonies. Then this is alpha complementation. This is the diagram. You can see that this is the plasmid. You can insert the foreign gene, then you can ligate the plasmid with the foreign gene, then you can transform the bacteria, and then you can select by seeing the blue white colonies. White colonies means it has the plasmid has the foreign gene, while the blue colonies, they are non-recombinant plasmids. Now we come on to restriction enzymes. This was the restriction enzymes, they were discovered by Nathan, Arbour and Smith. They were awarded the Nobel Prize in 1978. A restriction enzyme is an enzyme that cuts double-stranded DNA at a specific recognition nucleotide sequence, which are known as restriction sites. Now I have told you there are three types of restriction enzymes: type 1, type 2 and type 3. Type 1 and type 2, they are not used in genetic engineering, but the common type of restriction enzymes are type 2 restriction enzymes. They cut within the recognition sequence. Now these restriction enzymes, they produce two type of cuts, either blunt end, means same, and or sticky ends or cohesive ends. Now as I have told you, this EcoR1 recognition site is a palindrome. You can see GAATTC. You are reading from the 5' end, then again read from again the 5' end, GAATTC. Type 2, they are the most commonly available and used restriction enzymes. They are composed of only one subunit, palindromic sequence. They do not require ATP, but they require magnesium ion as a cofactor. Now these are the restriction enzymes, type 2 restriction enzymes. You can see some are producing blunt ends, some are producing sticky ends.